Alright, so hello, my name is Jared Fisher, and today we're going to be talking about the epiphytes presentation that I'm doing today, which is basically the research that we have been doing in our genetics lab for the past semester. So to start off, what are epiphytes? Epiphytes are organisms that are on the leaves on the outside of a plant and either have a commercialism relationship or a mutualistic relationship. And what that means is they either get benefits from the plants and don't benefit the plant at all, but they don't harm the plant at all, or if it's mutualistic, they both benefit the plant and they also benefit the organisms themselves. So you might be wondering, why are we so concerned about these epiphytes? And the main reasoning why is because we are trying to study to see if these epiphytes can affect heavy metals in the plants by breaking down these heavy metals. Now you might be wondering, why do we care about heavy metals? Well, heavy metals are heavily toxic to plants, and if you see in this graph, they can cause oxidative stress to plants. They could cause necrosis, chlorosis, and crinkle leaves in the plants. They could disrupt the photosynthetic machinery. They reduce the fruit quality. They cause squishing of heavy metals from subcellular compartments. Um, they cause modulation of the antioxidants, and they change the biochemical pathways. So they're very, very negative to the plants. But we're trying to see if we can get these epiphytes to break down these heavy metals into nanoparticles, because in their nanoparticle component, they don't cause as much negative impact on the plant. It's only when they're in their bigger molecules, when they're not in the nanos, that it actually causes a negative impact on the plant. So the way we're doing this is we're going to study them at Superfund sites. And Superfund sites are basically these sites that have been contaminated with these chemicals, sometimes through like a coal factory or sometimes through like a chemical spill that are required to be cleaned up by the EPA. And so typically the EPA helps clean it up, and the company that caused this or whatever organization caused this have to also clean up this area. But typically these areas are heavily contaminated with chemicals and heavy metals, and essentially they're contaminated with the stuff that we don't want to get influenced by the plants. So what we did is we took these plants from these Superfund sites that are actually growing, and we're studying the epiphytes on them, because if these plants are growing in these areas with heavy metals, then maybe the epiphytes, the bacteria that are on these microbes that are on these plants, are actually breaking down these heavy metals and causing these plants to grow. And if so, then maybe we could take these microbes, these epiphytes that are causing this breakdown, and we can cause an increased concentration of them in this area to help break down the heavy metals faster in these Superfund sites. That way, these Superfund sites can clear faster, and also it allows the plants to grow there. So, like, if we could do that for our crop, then we could actually use these Superfund sites rather than them just being these designated zone where nothing can happen. Now, I want to point out on this methodology, we really started right around here. All the other stuff, sample collection, the sample processing, even the mixed microbes were already done for us by a previous group. So we kind of came in this semester midway through the experiment, and so we did the second half. So the previous class basically, well, the TA collected the samples, but then the previous class did all the sample processing. They did some identification, but they basically did all the sample processing. And so we started off sort of with this, but like with those, but like with the just microbes with like a sample 1 through 12 for me. And so then we streaked this on a plate, and then it got amplified over here. But then from there, we actually have additional steps over here. So what we did is we also put it into like a glycol solution, and then we also put it in a solution to see if it had nitrates reductase properties. And then if it did have that, We then put it in an additional solution to see if it has silver reductase activity. And if it did, then that means it's breaking down heavy metals. And then from there, we put it in here. We basically did PCR, and then for PCR, we amplified it with the PCR. Oh, we lysed it, we got the DNA, and then we put it through PCR amplification, and we amplified the 16S DNA, and we then put it through Sanger sequencing. And then from there, we put it through the blast system to see what microbes were matched to the DNA that we collected. And then by matching those microbes, we could basically say, okay, well, these microbes that are having the silver reductase activity are the microbes that could break down these heavy metals contaminants. And so we might want to increase the concentration of these microbes, assuming they're not toxic to plants or humans. And so, like I was saying on the experiment, this one right here is the nitrates reductase reaction. And as you can see, my 2, my 6, my 8, 9, 10, 11, and 12 all had all tested positive. My 2, 4, 6, 8 through 12 all tested positive for the nitrate reductase test. And it's a little bit harder to see here, but this row is my sample. And if you see on this row, my sample 2, my sample 4, sample 6, 8, 12, 11, and 12 all tested positive for the silver reductase test. So then we put those through the PCR test that I talked about earlier, and we basically put it through all. We got the Sanger sequencing, we found the DNA, and then we um, put it through blast, and we found that both sample 6 and 11 were the same microbes. They were both Plantoa anthropelia, while 8 was Plantoa dispersa, and 12 was Pseudomonas rhizospiraeus, while 4 was the Hydrocarbophia. And two of the sample 2 right here couldn't be identified, so we put it through blast, but blast found there was no bacteria that's been archived with that DNA. So maybe it's an unknown bacteria, or maybe it's just didn't have proper sequencing. Which, if it didn't, that's okay because we still got some other ones. Really, we'll want to focus more on these two, because these two have been found to not be toxic to humans or to plants, but the Plantoa species are toxic to plants. But to conclude, by increasing, like I've said, by increasing the number of epiphytes that can break down these heavy contaminants, we can effectively reduce the time to lower metal contamination in these Superfund sites. Plus, we can also have crops growing in those areas. So that's once again effective use of that area. Acknowledgements. I would like to thank Professor Mukhtar and the graduate TA Binob, and also the biology department for giving me the chance to aid in this research. I would also like to thank the previous genetic lab students for the samples that they have worked on, because I would have had to start from step 1, then we would have been able to finish it. So we still have to thank them for giving us that foundation to complete the second part of this experiment.